episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest today who is helping to create a better tomorrow for all of us. Today we have the honor of being joined by Dr. Stephani Otte, who is Science Program Officer of Imaging at the Chan Zuckerberg Initiative, who leads their organization's imaging program and is focused on creation, dissemination, optimization, and standardization of transformative imaging technologies. Prior to Chan Zuckerberg Initiative, Dr. Otte was the Director of Science at a neuro-technology microscopy company, Inscopix, involved in accelerating brain science and innovating with some really cool miniature miniscope microscope solutions for real-time mapping of the human brain and its circuits. Dr. Otte received her Ph.D. in Neuroscience at the University of California, San Diego. She did her postdoc work in systems neuroscience at Salk Institute and University of California, Berkeley. When she's not doing all this cool stuff, she relaxes with hobbies like riding camels and skydiving, and we'll get into that in a little bit. And the Chan Zuckerberg Initiative is an organization that's established and owned by Dr. Priscilla Chan and her husband, Facebook founder Mark Zuckerberg, ultimately with a focus on science, education, immigration reform, housing, criminal justice, and other local issues. And their mission is to build a more inclusive, just, and healthy future for everyone, and to advance human potential and promote equality in areas such as health, education, scientific research, and energy. A lot to discuss. Dr. Stephani Otte, thank you so much for taking the time to come on the show.
Yeah, thank you. Excited to be here.
Yeah, it's, reading your background is quite fascinating. I'd love to start off, as we typically do, just by handing you the floor for a few minutes to talk a little bit more about yourself, a little bit of your background from where you grew up, when you developed these interests in biology and psychology and neuropharmacology, and a little bit of that early career journey. I think that would be a great way to get things started.
Yeah, happy to. So I kind of always had this fascination with the unknown, the unanswered questions. I was always a pretty inquisitive kid and just naturally got drawn to biology, mainly because the number of questions that were unknown. So I remember high school biology class just asking a number of questions and essentially saying, well, we don't know that yet. We need to study that. And so I was like, oh, this is what I'm going to dedicate my life to, is answering the unknown. The questions that really fascinated me the most had to deal with understanding of complex biological systems. The idea that I could have an input to the system and I get a different output depending upon the system state. Like a system can learn and it can grow and it can adapt and change and modify its responsiveness. How does it do that? What's responsible for that was where I was really intrigued. And so I think through my academic career, I took a lot of paths through biological systems. I studied the inner workings of cells, trying to understand protein interactions. I looked at neurons and their responsiveness, trying to understand how they encode information, then moved on to neural networks to understand how cells interact with each other. Yep. And it was during this time that I think I came to the two major conclusions. Number one, that to really understand complex biological systems and how they adapt and change was going to require much better tools than what we currently had. And two, that there was absolutely no way that I could do this alone. And so the idea of, instead of focusing on building out a research program and an academic career, I moved to focusing on tool and technology development and thinking about ways of accelerating the field became the focus. So I moved on, to do neurotech industry. After that, building out technology platforms to understand neural circuits. And that was really fascinating because it was like building the platforms with engineers, with computational groups, building out a number of different teams. So we're involved in building and dissemination of these platforms to world-renowned researchers across the globe. And then I still wanted more. I still realized that we had to understand different levels of investigations, then moved on to this Chan Zuckerberg initiative to run imaging programs. That's kind of a little little journey through my path.
Awesome. Awesome. And so a couple of years ago when Chan Zuckerberg initiative was announced, sort of this major sort of moonshot goal, cure, prevent, manage all diseases by the end of the century. And then obviously the recent announcement with focused on what you're doing with the AI imaging and other techs, $3.4 billion over the next 10 years. And once again, an important sort of mission, developing science and technologies to measure human biology in action. Obviously, if you're going to cure, prevent, and manage, you got to see what's going on. Talk a little bit about sort of CZI in general, and then sort of the imaging program within it, and also introduce us to the Chan Zuckerberg Institute for Advanced Biomedical Imaging.
Yeah, happy to. So as you say, the overall mission of the organization, especially the science part of the organization, is cure, prevent, manage all disease. And the idea here is you don't go one disease at a time. We're never going to get to the goal of doing this by the turn of the century by focusing in. We have to build a better understanding of complex biological systems. We need to accelerate the pace of scientific discovery. We need to invest in tool and technology developments. So this is what CZI science as a whole is focused on. As part of that, you have different levers that you can utilize to help drive the field forward and drive progress forward. So you have a granting arm that allows you to give money to individuals or organizations. You're seeding funding, exciting research, figuring out ways of having groups work together. We also have the ability to build. So we have a software development team in-house that acts as a professional software development team that can build platforms that can be utilized by the broader scientific community. And all of our science platforms and software development is all open source. And a lot of times we're inviting the community to build with us. And then recently, we still notice that there's this major gap within the ecosystem. So you have lots of money going into discovery research, and then you have companies on the other end that are looking to scale platforms and broad distribution of the platforms. But there's this point in the middle where you're really trying to integrate a number of technologies together. So in imaging space, a lot of times you need hardware platforms, the computational tools, the software needed to both visualize and analyze. And sometimes you have to do like biological probe engineering so you can actually see the thing that you want to see. And most of the time, all of these things have to come together to actually enable an application to actually see a biological system. And academia is not well set up to do these types of pursuits. Industry, it's a little bit too risky to put that much investment in some of the early stage, in the early stage work. So what the Institute will do is kind of sit at this hybrid between academia and industry, working on more collaborative projects as like a system engineering approach to be able to advance promising imaging technologies. So sort of set up its own culture, but be really connected to the community. So connected to our grantees, connected to the broader ecosystem. And so we're early stages of development here. So we got approval for funding. And now 2022 is going to be a build year for us. So we're installing leadership, securing location, building everything out. So that's the focus right now.
Excellent. Excellent. And, I was looking at sort of the 2020 numbers in sort of the general bioimaging sector. It's, close to $60 billion, it seems, globally. What are some, when you think of imaging, what are some of your holy grails in this space? Is it about seeing what we currently see a lot clearer? Is it about seeing smaller? Is it about cheaper, more portable, all of the above? Take us to sort of what the big wishes are. If you can snap your fingers tomorrow, have like the best tools available. What are these holy grail possibilities in imaging?
Yeah, right now we have pretty limited view into complex systems. Okay. I can't see inside the cell to a high degree. I don't know how the proteins are interacting inside the cellular environment. I don't know how a virus gets in and hijacks normal cellular functioning for its own propagation. We don't have a view on this, and it's a complex process, and we can't really see inside the cellular environment with high resolution. Okay. So I think being able to open up new windows inside the cell is very exciting. I also think opening up new windows of actually seeing cells and their signaling process working collaboratively together inside a living system is also a very exciting area. So like deep tissue imaging, right now, a lot of people, like microscopy is only going to penetrate so far into the system. It's basically a black box, and it's very low resolution with what we can see right now. But there's a number of tricks that are being applied, a number of different imaging modalities that are being tried to actually get high resolution imaging inside the cell, or inside the body. You could imagine like watching immune cells attack cancer cells. If you're doing drugs, like how is the system behaving? If you take out a tumor, are there remaining cancer cells? Where are they? How are they interacting? We'd love to be able to open those new views into biological systems. And then there's the idea of actually being able to integrate across levels. So to actually understand the system, it's not just the cell is operating in its own, it's operating in the context of a complex system within the tissue. So being able to connect what's happening at the whole body level, organ level, tissue level, cellular level, inside the cellular level, and being able to pull some of these threads out to actually understand across scales what's happening with complex biological process are areas that I am definitely interested in. I think when we talk about development of those approaches, the only way that we're actually going to be able to understand these systems is actually getting the technology in the hands of researchers and allowing them to make and build on each other's discoveries and making sure that they can actually not just see the thing, but understand what they're seeing, be able to quantify it. So building out tools and technologies to allow for like quantitative analysis and making sure that those are cost effective, can get into the hands of people, and that we train them to do it is also a major focus of the initiative. So it's both the development and dissemination of advanced imaging technologies.
Stephanie, when we do the show, obviously we'll talk about certain disease states and sort of the typical imaging tools that we all have from our own time in sort of the medical domain, pop up, x-rays, PET scans, CAT scans, ultrasound, of course. Went online to try to do some research on the stuff that's coming, ran into a lot of really interesting, cool sounding things. Not sure what they are. Terms like multispectral optoacoustic tomography, all sorts of interesting things about novel families of dyes, reversible photoswitchable protein technologies. Take us through sort of some of the areas. You don't give a lecture on sort of the next 10 years of what's hot, but what are some of the things that are hot? Some of the areas that excite you, and what does some of this stuff mean?
So I think there is a lot of interesting things happening in what we're calling deep tissue imaging, being able to visualize at high resolution inside the body. And there's a number of technologies being pointed at that. So from photoacoustic imaging, which is a combination of light and sound, which should allow the light can penetrate, it's getting the signal out. So we want to be able to, it's basically combining lights and ultrasound at the same time. And to do this, you also need to develop biological probes. So some of the things that you're listening, you actually are, that you were noting, we actually have to label the thing that we want to see. So we do some type of genetic engineering to actually be able to see the molecule or see the thing. In that deep tissue imaging space, there's like X-ray tomography approaches that are pretty interesting, that this allows you to get higher resolution, large volumes. Right now it's in postmortem tissue, but has the possibility of moving into living specimens, which would be quite interesting. Imagine getting like full visualization of an entire organ down to cellular level, and being able to see both the ultrastructure and what's going wrong and disease states. So I think that's a really exciting area. And then there's just some, interesting stuff that's like very early, like quantum imaging and, and other things, where it's just being able to, without going into technical details, basically new approaches to see things in greater detail that we've never seen before. So we can either go smaller, wider, deeper, or faster, essentially. And there's a number of techniques being developed to do that.
Very cool. Very cool. And then alongside sort of the imaging component of this new initiative at the Zuckerberg initiative is the term artificial intelligence, which is something that's popping up now everywhere. We talked a little bit about it actually with folks from Microsoft last year in terms of how it can supplement, be used in conjunction with the clinician that's analyzing this data. But now you're getting into a lot more data, a lot more resolution, a lot more, whether you're talking about proteins and cells and tissue structures and everything else. Talk a little bit about the benefits of artificial intelligence, machine learning, some of the synergies that you see with some of these novel imaging technologies.
Yeah, you can imagine some of these data set sizes get incredibly large, especially if you're looking at high resolution dynamic imaging. I'm taking high resolution snapshots regularly of a complex system. And then you have to start picking out the features that make sense. What am I trying to pay attention to? There's so much data. So a lot of times, with imaging, just picking out segmenting the thing that's important and being able to watch it over time. The way that we used to do this is, grad students circling things. And it was very laborious, took a ton of time and a ton of effort. And so being able to automate some of these processes, show what's important, be able to identify what's important and be able to automate the entire data stack so you can easily get out the features that you care about. That is quite important. Speeds the field up dramatically and can improve things both in time and effort and our understanding. So I would say that's a big area where AI and ML is being applied in imaging, but you could apply it even in acquisition as well. So imagine that you're trying to acquire something about a system, but there's only certain points in time that you actually care about where the thing is actually occurring. You can actually have smart acquisition systems that know where to focus, what data to collect. So you can also minimize data collection on the front end. So I think it can be applied basically at any stage in the processing, all the way to actually analyzing the systems and starting to pick up patterns at the end about how they're behaving or how they're interacting.
Stephanie, you have so you spent a fascinating time before the Zuckerberg initiative looking into brains with these fascinating mini scopes. Obviously, you have a background in neuropharmacology. You like the CNS. Talk a little bit about some of the obviously, these tools can go in all directions, as you're mentioning, the tumor microenvironments and so forth, if drugs are getting where they need to get and so forth. Some interesting pathologies, disease states that you're personally interested in seeing the movement of some of these new tools in the coming years.
Yeah. Well, I definitely have an interest in neuroscience space. So, and it kind of goes back to the, understanding of complex systems and adapting, changing. So I think for a lot of systems like addiction, for instance, like understanding addiction circuits, understanding how the brain can get into depressed states or manic states, understanding even neurodegenerative illnesses or circuit-based disorders like Parkinson's disease. There's a lot of really interesting things that end up being, it's a complexity of interactions that are causing the system to go awry, that it's now in this disease state, and being able to understand that, because it's not going to be like a simple causal one gene created this. It will never be that way. And so we need to understand actually how the system is interacting, build better theories and models of how to predict the system state, and then how you can influence it when it moves into a disease state. So I really like thinking on those level. Right now, I'm not doing as much neuroscience, but I'm hoping, in the future that more of these neuroscience tools will be created as well to study the brain in greater detail.
And, you have that interesting paper from a couple of years ago where you were looking at, sort of the optogenetics when you were using some of those tools with the meniscopes. And it just got me thinking, because we did an episode about a year ago with these folks from the U.S. Air Force on this new system they call iNeurals, where they're not just sort of looking at interesting images in the brain when the airmen are learning to do something, but then the reverse, sort of what I'll generally call the zap, how we can move things around or influence. You studied optogenetics in the past. What do you think about sort of the combination of some of these new tools, not just as a diagnostic tool to see things, but also a therapeutic at the same time?
Yeah, I think a lot of times when you're pushing imaging technologies, this is exactly the thing that comes right afterwards. So with imaging technologies, we had green fluorescent protein and could now label things to be able to visualize them. And so there was a number of genetic tricks in terms of how we could put this into cells, how we could visualize them over time. And then you go into the idea of, okay, now I have an understanding of the system. I can see how it operates, but how can I perturb it to make it operate in a more beneficial way? And so a lot of times sometimes you're using similar genetic tricks to be able to put mechanisms of control into the circuit. So optogenetics is kind of the flip side of that. And you can use all optical approaches. I can now have a read-write on the system and see how it's operating. I think that the operation, a lot of these are in like preclinical models, not necessarily used for medical purposes. So I think as you move into that space, you have to think through ethical implications, and it's important to think through perturbations of the system is an important thing to think through thoroughly.
Absolutely. Stephanie, you wrote an article a couple of months ago. It was entitled "Why Sharing Bioimaging Technology is Crucial to Global Health." And this, you talk about a program expanding global access to bioimaging, that's part of the CZI initiatives, looking at how we can get some of this imaging expertise, different areas in Africa, Latin America, the Caribbean, and so forth. Talk a little bit about this program, if you would.
Yeah, so this is part of the dissemination aspect of the imaging program. So we deeply believe that imaging technologies can provide a better fundamental understanding of disease states and biological systems. And then if you look at distribution of imaging technologies, who's using them, what they're using them for, and think about global health burden, there's a huge discrepancy. So most of the imaging technologies are in places with the lowest health burden, and the places with the highest health burden have low access to these technologies. And so what we're trying to do at CZI is create a global network of imaging scientists. So these are people that run imaging core facilities. Their job is to make sure that the researchers around them have access to imaging technologies, that they know how to use them, that they know how to analyze the data. So we're supporting a global network. We're encouraging collaboration. We're getting people trained up on how to use them. And we have found that when governments or universities are investing in infrastructure, core pieces that allow all research to be better, that there is significant improvements in the research that's being done. And so we would like to see this also in developing regions, and thinking about how to get their infrastructure updated to make the most use of their science. So we expanded our program to include Africa, Latin America, former Soviet countries, so Eastern Europe, and we'll continue to expand. And we're integrating them in the global network, getting them up and operating faster, getting them access to equipment and training.
How do startup innovators work with you? I mean, I know that you have a lot of, obviously, the relationships, and you're putting a lot of resources into some of the larger institutions. But if I'm a company of two with really cool imaging technologies, or a postdoc at a lab, and I have a really good idea, do I come directly to CZI? How does that work? And what's sort of the process with working with sort of the smaller innovators?
We haven't supported smaller innovators
Okay.
to date, but this may change
Okay.
in the future. So we have the capacity to do it, but we want to make sure that we're doing it in an equitable way, thinking through distribution of the technology and what to support. So as we build the Imaging Institute, we will be thinking through this in terms of off-ramps of the technology. So one thing CZI will never be involved in, or I should never say never, but like the distribution, customer support, manufacturing, especially hardware devices, this would not be something that we would consider at this point. And so working with partners that can scale, working with people that want to take it into the commercial space is important, but we want to ensure that there's some value alignment, meaning we want to keep, costs reasonable for maximal distribution. We would like some customizability on equipment too, just to make sure that the field continues to accelerate and it doesn't get closed off for further innovation. So we're discussing best ways of doing that right now. Most of the time when we explore the space, we talk with a number of people, try to understand what win-win situations would be. How can we work together? So I would say it's possible something like this is to come, but we're not doing it currently.
Stephani, you've had a fascinating journey so far, academia, private industry, and now this foundational work. Take a couple of minutes, if you would, just to talk about some of the important mentors that you've had along the way, the folks that have influenced you, that have sort of kept you on this path when, in the good times, but also the times where you might have said, I'm not doing this biomedical stuff anymore. I'm going to get involved in an entirely different area.
I think mentors have been critical through all of this, and I'm taking kind of a interesting approach through science, not just like the typical path. And I would say the way that I view this is I try to view everybody as a mentor, even when the relationship may not be the best. It's like, what can I learn from this? What can I take from this? How can I learn and grow and develop from this? There's lots of people that were, from undergraduate, graduate school, postdoc, at industry, now at the foundation, like people that I look up to a ton. And then there's also people that, I've learned a lot about myself, how to be resilient, how to work through problems, even through little conflicts, we can learn this as well. But I think mentorship is critical. And I think especially it's important to find those mentors and then mentor for other people too, that are taking different career paths too. I think a lot of times in the academic environment, they sometimes, it was, they used to talk about the only path, it's like, this was the one path to take. And so having people that also support exploration of paths and moving across industries and sectors is also really important, and finding people that have done that as well as sources of inspiration.
Flipping that around, the next generation that's listening to the show, that's just headed off to college for their freshman year, or maybe in college trying to figure out what they're going to be doing. Tell them why it's a hot time to get involved in imaging sciences in 2022.
Oh yeah, I mean, I think that this is a really starting kind of a revolution in imaging science space. There's a lot of money, a lot of focus going into this. I think this is focused on tool and technology development, and then the broader application of those tools and technology, a little bit less focused on like a deep dive specific research question. So I think that there's going to be more interesting opportunities available. I think a lot of people like working more in collaborative environments on project-based science, of thinking through larger impact of their efforts. And I think there's going to be a lot of these opportunities becoming available with a lot of experiments and the way to conduct science happening. And I think that they can stay closer to the discovery, but also think through broader impact. And these are things that can help both our understanding of, biological systems, and then potentially in the, curing, preventing, managing disease in the future.
Excellent. Steffi, final question. Are you allowed, now that you're at the Chan Zuckerberg Initiative, to skydive or ride camels still? And if you are you getting much of that done during the pandemic? And last part of that, have you ever dove into the desert and jumped on a camel? And then I was thinking about that when I was
No, I would say this was the 20s version of myself that was very adventurous. I did a lot of international travel backpacking trips during that time. Now I'm a mother, and I think less of Chan Zuckerberg Initiative, but more my husband and children would probably not want this to be something that I focus on now, but still really like travel, exploration of new cultures. Would love to do that more, but have been sort of in the pandemic phase for the last 2 years.
Gotcha.
Pretty stationary.
Gotcha.
Yeah.
Well, hopefully you can get back to it one day, because it sounds pretty cool. But
yeah.
So it's a fascinating set of programs you have there, fascinating journey you've been on, and really rooting you on as you build this up for the organization and it develops. So it's really exciting. Any final messages for the audience? Anything I missed that you might want to mention? Please, I'll give you the floor on the way out.
Yeah. I don't think so. Yeah, I just think it's a really exciting time for imaging technologies. And then, we're always open to ideas and creative ideas and thinking through ways of really accelerating science. And I think that's important to do, and will require the collective effort of a lot of minds. We'd also love new people into the field too. So bringing more people into this field, opening up jobs and opportunities will be quite exciting.
Excellent. Well, for everybody that's going to be listening to this episode on our podcast networks or watching on the YouTube channel, once again, you've been listening to Dr. Stephani Otte, Science Program Officer, Imaging, the Chan Zuckerberg Initiative, doing amazing things to develop science and technologies to measure human biology in action, ultimately towards their main mission of curing, preventing, or managing all diseases by the end of the century. Stephani, I want to thank you for taking the time out of your schedule to come talk to us for a little while. Thank you for everything you've been doing there at Chan Zuckerberg. And as we say on this show, thanks for helping to create a better tomorrow through all the research you're doing. Really fascinating story.
Thank you so much. Really appreciate it. Enjoy my time.